I'm Ranga Deshpande. Hello, everyone. I am from Auburn University MRI Research Center and currently a postdoc at UCLA. Um, okay, so as the title says, this work is on brain network of emotion regulation in soldiers with trauma. This has implications for studying emotion regulation as in general and dysregulation in soldiers. So the outline is pretty simple. Background data acquisition, the task, the emotion regulation task that was performed, dynamic effective connectivity modeling, that's the trick that we used, and results and conclusions. Yeah. Little background that all of you might be aware of. Emotions arise when there is appraisal, when we assess a situation as good or bad. And as such, when something something is unfavorable to the organism, there will be negative emotions. Hence, there is an inbuilt mechanism in the brain to counter such situations and enhance survival that is called emotion regulation. And because it is such a complex mechanism, so many mental disorders show dysregulation of emotions, which then compound onto several symptoms. So, coming to emotion regulation and dysregulation, a lot of work has already been done. A lot of credit goes to so many researchers that have contributed. And people have kind of identified which brain regions might be involved in emotion regulation in general, irrespective of the psychiatric disorder. And that's the consensus is the dorsolateral prefrontal cortex or the middle frontal gyrus, the insula, the medial prefrontal cortex, and amygdala. These are the regions that usually pop up in activation and connectivity studies. However, what is the origin of emotion regulation in the brain? And what is the network of information flow? How does information flow from where to where? And what is probably the cause? And who are the mediators? And what are the effects that are happening in the brain during emotion regulation? That has not been entirely understood, and a lot of work is going on to decipher that. And this work is a step in that direction. And I'm really interested in the cause, to find what is the cause, because if we are able to identify the cause and do something about it, the mediators and the effects would take care of themselves. So our approach to do that was to get the brain network that is involved in emotion regulation. So in this work, we identified directional brain networks of emotion regulation in healthy participants and the dysregulation in soldiers who had comorbid PTSD and mild traumatic brain injury. We had 59 male soldiers who went to Iraq and Afghanistan, 36 comorbid and 23 match controls. We did scanning in a 3D MRI scanner at Auburn, and we had a pretty good TR of 600 milliseconds that would help us in dynamic modeling. We did standard pre-processing in SPM. I have a poster 1389, so you could drop in if you want more details about that. So the emotion regulation task, which is central to this study, involves soldiers viewing military-specific imagery because that is relevant to them. And they would see certain neutral images, certain positive images, certain negative images relevant to their experiences in Iraq and Afghanistan. And so as the task goes, they would view an image for 3 to 7 seconds, and then that would actually elicit an emotion, either positive or negative. And then they would either be asked to maintain that emotional level, or suppress that emotional level. So naturally, when they are suppressing the emotion compared to maintaining the emotion regulation is happening, and that is where we would gain our contrast. So I'll just, we use military imagery. I'll just go through the task so that you understand a little more about how this was done. So there is a fixation cross. They would view an image. In the maintain, they would just maintain that emotion that was generated in them. And then they would view another image. This is actually a negative image, which would elicit negative emotion. and they would be asked to suppress that emotion, which would engage the emotion regulation circuitry. So once we have the data, we wanted to first identify brain regions which are activated during the emotion regulation condition, and we used a pretty conservative approach wherein we did GLM analysis to find activated brain regions, and we also took the first level maps from each participant with a liberal threshold, and we also took the activation maps from a meta-analysis and did an overlap of all the maps, and then finalized on our brain regions because we wanted this to be pretty conservative and strict. So the brain activations and the final ROIs that we determined from GLM analysis were pretty much as expected in any emotion regulation study, with the MFG stands for middle frontal gyrus. So middle frontal, the medial frontal gyrus, the amygdala, and the parietal regions and insula. These were the regions that popped up. So next what we did is dynamic effective connectivity modeling that really enabled us to get the brain network. So as the word says, it is effective connectivity modeling wherein we get directional causal relationships. We used a Granger causality framework because it was not completely model based. And dynamics says that we get connectivity value at every TR. And this is not a window-based approach, so it is not a low-pass filtered version. It is based on a dynamic Kalman filter, so we get dynamic connectivity values at pretty high resolution that will enable us to estimate the connectivity. So in this figure, I'll explain how we got the actual connectivity relationships. Now looking at the maintained condition, we had 5 TRs. So we have 5 connectivity matrices for the maintained condition for the entire, all the ROIs. We took them and we averaged across the TRs. Then we took the suppressed condition. We get connectivity values for all the TRs, and then we then average them out. So now we have maintained connectivity values and suppressed connectivity values for controls and the disorder group. Next, what we did is to get the regulation network, we consider only healthy controls, and we compared the suppressed condition with the maintained condition. So suppressed greater than maintained is emotion regulation. Next, for emotion dysregulation, we just took the suppressed condition because that is where the emotion regulation is happening. And we did a comparison between the disease group and the healthy group. So this is the network. I'll show a neater figure in the next slide. This is the network that we got, the actual statistics map, T-statistic map. There was a similarity of 88% between regulation and dysregulation. And before we move further, it's important to understand what these represent. The emotion regulation network gives the strength of connection when regulation is happening. On the other hand, the dysregulation network gives the magnitude of imbalance. It is not an absolute value, it is a comparison. So it tells how much there is an imbalance during dysregulation compared to the regulation condition. So this shows the emotion regulation network that we found in healthy controls. I split up the network into 3 subnetworks so that we can understand it better. The first one is the direct influence of the middle frontal gyrus on amygdala and angular gyrus. So when you see this figure, try to imagine the cause, mediators, and effect, that relationship that I showed earlier. In the second figure, the middle frontal gyrus is having an influence on insula and anterior cingulate, which is then having an influence on the amygdala and the angular gyrus. And in the third figure, there is a secondary relationship between insula and the superior temporal gyrus. So to get a quick sense of this, you can consider the middle frontal gyrus is possibly the cause because it is at the top of the ladder. And we can consider the insula, the anterior cingulate, and the supplementary motor area as the mediators because it's in the middle of the ladder, and the amygdala and the angular gyrus as the effect, which because it is at the bottom of the ladder causing and this fits well with the behavioral manifestations we observe with emotion regulation. Next, coming to dysregulation, please note here that blue is lower connectivity and red is higher connectivity. So we see that in the connections arising from the prefrontal cortex have gone down in the disorder, and connections in the subcortical regions have gone up. And this, it is quite evident that if we take this network literally, there is a reduction in regulation from prefrontal regions, which is leading to an overdrive in the subcortical and parietal regions. And this fits well with what behavioral manifestations we observed in the soldiers that came to the scanning. So just comparing these two, this is just a comparison of the two. So to wind up, the regulation network has broad relevance for understanding regulation mechanism in healthy and diseased brains. And the dysregulation network kind of gives a feel of how it might be the case in other disorders which have dysregulation issues, and how it might be in PTSD and MTBI. And this work was done at Auburn University. I'd like to thank my advisor, Tom Denny, who is here today. I'd like to thank my collaborators, Gopi Deshpande, Jeff, Mike from the Army, and Alex, and everyone from the MRI Center. Thanks.
We have time for one quick question.
Thank you for the presentation. Did these individuals receive any training on how to perform the emotion suppression?
Yeah, there was a dummy session before the scan where they were just presented a quickie, and so that they understand the instructions and they know how to respond when the instructions are given.
I mean, I just mean in terms of suppressing emotion, like what were they asked to do?
Yeah, they had to come up with cognitive reappraisal methods of their own. Maybe think of something happy in their life, something that they can think of, it's basically cognitive reappraisal. Think of something that will make you feel better.
Okay.
I just had two quick questions. The first one, a lot of the regions you showed seem to overlap with depression. Did you screen any of the subjects for depressive symptoms? The second question I had with the depression of the connections, did you screen anyone for beta blockers? Because you do see that if they're taking some medications for stress or depression.
Yeah, so we screened out everyone who had schizophrenia, depression, and any related psychotic disorder. And as well, none of them were under medication. Yeah, so anyone who's interested can drop in at 1389 this afternoon.
In order to interpret causality, did you take into account the fact that the hemodynamic response is different for different regions and different subjects? There's a very strong heterogeneity that is really it's a strong confound in order to evaluate things like granular causality in fMRI. So do you account for that, or do you?
Yeah, so we actually perform deconvolution on the data at the pre-processing stage, which will technically remove any impact of the HRF and estimate the neural signal. So it will improve the Granger causality estimation.
Okay, thank you.
Thank you.
Could you please say something more with your task? That is, you use emotion suppression task, but you know the suppression can be very broad or very narrow. Which concrete strategy do you use for the suppression? Could you please say something more?
Yeah, so the instructions were kind of pretty broad. They were not asked to do anything specific, like do this or do that, but they were asked to come up with any way that they do in normal day life to help them feel better. So they were asked to feel better when they were asked to suppress.